Chers et chers collègues, chères étudiantes, chers étudiants, mesdames, messieurs, je suis extrêmement heureux d'ouvrir la conférence Marc Bloch de l'École des hautes études en sciences sociales et de le faire devant des descendants de cet immense historien dont les analyses, fortes et lucides, résonnent plus que jamais dans notre monde instable. Je me réjouis également de prendre la parole dans les magnifiques locaux du Musée de l'Homme et je remercie sa directrice, Aurélie Clémenté Ruiz, pour son accueil. Dans le cadre de cette 42e édition, je souhaite la bienvenue à Svante Pääbo. Après des études à l'université d'Uppsala en Suède, à la fin des années 70, Svante Pääbo a démarré une brillante carrière universitaire qui l'a conduit à l'Institut Max Planck de Leipzig. En 2022, son œuvre scientifique a été couronnée du prix Nobel de médecine. À celles et ceux qui s'interrogent sur la présence d'un médecin dans notre institution de sciences sociales, je rappellerai les venues dans le passé pour cette même conférence d'un prix Nobel de physique, Ilya Prigogine, et d'un grand biologiste, Jean-Claude Amezen. À relire leurs conférences, et avec le recul du temps, je suis surpris de les voir aborder des questions toujours majeures pour les sciences sociales. En 1987, Prigogine nous invitait à une profonde modestie dans nos rapports disciplinaires au savoir et surtout à éviter toute hiérarchisation dans les questionnements et les disciplines. Après tout, nous disait-il, l'histoire et la physique, par exemple, ont beaucoup en commun, à commencer par des raisonnements contrefactuels ou prédictifs similaires. En 2006, Jean-Claude Amezen construisit, lors de sa conférence Marc Bloch, son propos autour des métamorphoses biologiques et sociales et des difficultés à stabiliser les catégories analytiques des mondes du vivant. Pour ne pas oublier nos métamorphoses, nous suggérait Amezen, ne les prenons jamais pour argent comptant et soumettons-les sans cesse aux exigences cumulatives et rigoureuses de la science. Tous deux nous invitaient à réfléchir aux frontières disciplinaires, en respectant les épistémologies propres aux sciences sociales et aux sciences expérimentales. L'interdisciplinarité trouve alors sa pleine mesure en nous permettant de sortir d'une hyperspécialisation stérile, de ce chaos disciplinaire dont parle le sociologue Andrew Abbott, en insistant sur les distinctions fractales provoquées par une trop lourde disciplinarisation. L'interdisciplinarité nous appauvrit également si elle n'est qu'une injonction bureaucratique à singer les épistémologies d'autres disciplines. En revanche, elle nous enrichit si elle ouvre des perspectives, approfondit nos méthodes et étoffe nos interprétations. Aujourd'hui, ce soir, Svante Pääbo est présent à nos côtés pour évoquer le cœur de ses travaux sur l'ADN et la discipline qu'il a contribué à forger, la paléogénétique. Alors que Prigogine et Amezen ont beaucoup débattu de fragmentation, de dislocation, Pääbo travaille sur ce qui nous unit à travers les siècles, la longue durée, chère à l'école, des hommes et des femmes du Néandertal et de leur patrimoine génétique. À travers ces recherches, Pääbo offre de nouvelles manières de repenser les dynamiques migratoires, les circulations et les causalités des épidémies, les frontières de l'humanité ou encore les stratifications sociales du passé. Pour ne prendre qu'un exemple, je me permets de citer les recherches en cours sur les migrations postérieures à la chute de l'Empire romain, croisant des enquêtes historiques et archéologiques à partir des possibilités d'identification offertes par la méthode qu'il a mise en œuvre. Au-delà de l'histoire, l'anthropologie, la géographie ou encore l'archéologie ont beaucoup à dire, à dialoguer, à débattre avec cette relecture des origines de l'humanité. Au-delà de sa richesse interdisciplinaire, la recherche de Svante Pääbo m'inspire une double réflexion, à la fois institutionnelle et scientifique. Tout d'abord, je tiens à souligner l'importance du soutien d'une institution, en l'occurrence le Max Planck Institute de Leipzig, qui n'a pas lésiné sur les extraordinaires moyens nécessaires à la mise en œuvre d'un projet aussi ambitieux. Je tiens également à souligner la richesse de son obsession de chercheur, une caractéristique toujours présente dans les grandes œuvres de recherche. Dans ce beau livre que je vous invite à lire, Néandertal à la recherche des génomes perdus, il ne cache pas les origines anciennes et personnelles, dès son plus jeune âge, de cette quête étonnante de l'ADN de l'homme et de la femme de Néandertal. Grâce à ce fort soutien institutionnel et à ses propres obsessions scientifiques, Pääbo a trouvé, après de nombreux échecs, dans une grotte de Sibérie, connue sous le nom de Denisovans, un petit fragment d'os permettant d'identifier le génome d'une nouvelle espèce. C'est dans cette grotte qu'il nous emmène ce soir, pour nous permettre d'ajouter un versant biologique à notre compréhension des sociétés humaines. Dans une enquête publiée de manière posthume, le sociologue Erving Goffman avouait à la fin de sa carrière qu'après avoir beaucoup travaillé sur l'environnement social, largement construit par la société, nous disait-il, il était temps que les chercheuses et les chercheurs prennent mieux en compte les questions biologiques. Le débat est donc ouvert ce soir au Musée de l'Homme, il est ouvert à l'EHESS, et je laisse maintenant, avec beaucoup de plaisir, la parole à Svante Pääbo, que je remercie très chaleureusement.
Well, thank you very much for that generous introduction and for the great honor to give the Marc Bloch Lecture here today, the first time after the pandemic, I understand. So what I then wanted to start out by doing is reminding you about what you are all well aware of, I think, that our genome, our entire genetic material, is stored in essentially every cell in our body on the chromosomes in the form of this famous DNA molecule. And as you will also know, this molecule, the information there is stored in the sequence of 4 letters or nucleotides, and it's stored essentially twice on each of the 2 strands. And that's important because whenever a cell will divide, those 2 strands will be unwound and 2 new strands will be formed with the old strands as a template. And that process is very exact, but sometimes errors are made. A wrong base is built in. And if that is not repaired before the genome divides again, it results in a mutation. And we can see the result of those mutations that have happened in the germline when sperm and eggs are formed in the form of differences in DNA sequences in the population. So if you compare 2 genomes of 2 people in this room, for example, you will find a difference approximately every 1,000 such letters. And since these mutations rain down on the genome in every generation, if you add a chimpanzee to this comparison, you will find more differences, approximately 1 difference every 100 such bases. And you can then use these differences to reconstruct the history of this piece of DNA you look at, and we generally depict that history in the form of one of those trees. And in this case, it's very, very simple, of course. The 2 human sequences have a common ancestor here, and further back in time, approximately 10 times further back in time, is there a common ancestor shared with the chimpanzee. And you also know that the genome have about 3 billion such letters. So there are about 3 million differences between any two genomes, so there is a lot of information there to reconstruct the genomic history of our species. And when you do that on a global scale, one amazing finding that came out very early on was that most of the variation among humans exists in Africa, south of the Sahara. And if you take all people outside Africa, there is less variation there. The reason for that is then that there is a sort of part of the variation in Africa that exists only there. And the interpretation of that is that the ancestors of everyone today, the ancestors of present-day humans, evolved in Africa, lived there for a while and accumulated genetic differences, and a part of that variation, so to say, went out and colonized the rest of the world. And with genetic tricks, you can also estimate when that happened, and it's rather recently in evolutionary terms. Less than 100,000 years ago or so, when this exodus out of Africa happened. But an interesting question is, of course, that 100,000 years ago, modern humans were not alone on the planet. There were other forms of humans there. And in Eurasia, very famously then, in Western Eurasia, Neanderthals, and in Eastern Eurasia, other forms of now extinct humans that we know less about. So Neanderthals then, on the left here, were these robust forms of humans that appear in the fossil record maybe 400,000 years ago or so, 500,000 years ago. They live in Western Eurasia then until they disappear about 40,000 years ago, in connection with that modern humans to the right here appear on the scene. And you may then ask why we should be interested in Neanderthals. And for me, there are at least two good reasons for that. One is that they are the closest evolutionary relatives to everyone around on the planet today. So if we want to define ourselves as a group from a genetic or biological perspective, it's them we should compare ourselves to in terms of how are we similar to them, how are we different from them. Another thing is, of course, that they were here quite recently, until 40,000 years ago or so, so maybe 1,500 generations or so. So you may ask what happened when we met, did we mix, etc. But to then ask that from a genetic perspective, you will need to retrieve DNA from bones that are at least 40,000 years old. And that work then goes back, the technology for that goes back to the early 80s, when we in Uppsala then started to try to retrieve DNA from something that's much younger, from Egyptian mummies that are 2,000, 3,000, 4,000 years old and look very well preserved, such as in this case here. And if you look microscopically in the skin of, for example, this mummy here, you can see the surface of the skin nicely, and in basal levels here, things that look like cell nuclei where the DNA would be stored. And indeed, you can stain these nuclei and show that there seems to be DNA preserved there. So I was young and very enthusiastic. I went on and extracted the DNA and sequenced down DNA and found some human DNA there and published it to great fanfare. And sadly, over the years that followed, it became clear that the DNA sequences I had determined there were clearly not from the mummy. They were from me or from some museum curator or someone else. So, and the reason for that is then that there are a lot of sort of technical issues with this type of work, it turned out. So what survives in old tissues is small amounts of DNA if you're lucky, but it's in the order of 100,000, a millionfold less compared to present-day tissues. It's also degraded to short little fragments compared to fresh DNA you would extract from a blood sample from one of you or so. It carries chemical modifications, and the vast majority of the DNA in any such specimen doesn't come from the individual in question, it comes from bacteria or fungi that live there. And the result of that is that tiny little amounts of human DNA on a dust particle in the room, for example, would not at all be of importance when you study present-day DNA, but may totally overwhelm the experiments when you look at these old things. And that's what then had happened to me there in the first experiments. So we worked over many years on developing methods to overcome all these different problems. It involves working in clean room conditions and lots of our other things. We did that mainly by working on extinct animals, where it's much easier to distinguish the endogenous DNA from contaminating DNA that particularly come from humans, and so ground sloths, mammoths. And once the technology became better, many people started applying this to extracting DNA from many different sources of old organic remains. But our interest was then always particularly on the Neanderthals. And at the time then, in the early 90s, there were two ideas about how Neanderthals would relate to present-day humans. One idea would be that modern humans come out of Africa, spread to Europe and Asia, and replace Neanderthals in Europe and other form in Asia with no mixing whatsoever, so total replacement. Another idea is that modern humans come from Africa, come to Europe, mix with Neanderthals in Europe, mix with other forms in Asia, so that there would be some special relationship between Europeans today and Neanderthals in Europe, for example. And you can imagine any kind of sort of gradation of this, from total replacement, 0% contribution from Neanderthals here, till actual total continuity. There were some people who thought that Neanderthals were actually the ancestors of present-day Europeans. So we were very lucky then in the early 90s to get access to a Neanderthal specimen, not just any Neanderthal specimen, but the Neanderthal that was found in Neanderthal in 1856 and gave its name to this group of humans. And I think that was lucky because paleontology is a difficult discipline, and almost any other specimen at the time we would have studied, someone would have said it's not quite a typical Neanderthal, it's a bit too gracile, or it's something wrong with it. But if this is not a Neanderthal, then Neanderthals don't exist, so to say. At the time, the technology allowed us to look on only DNA that occur in many copies per cell. So one focused on this mitochondrial DNA located outside the cell nucleus that exists in thousands of copies per cell. And we then retrieved a small part of that DNA, reconstructed it, and could compare it to the variation in present-day humans. And for this mitochondrial DNA then, for present-day humans, they all go back to common ancestor 100, 200,000 years ago. And the Neanderthal mitochondrial DNA fell outside of that variation, went back to common ancestor more than 500,000 years ago, so it looked very different. After that, we looked at many other Neanderthal remains. They all fell together outside the variation of modern humans. So in this scenario then, for the mitochondrial DNA is total replacement. There's no one walking around today with a Neanderthal mitochondrial genome. But it was very clear, of course, that the big genetic history, or the more complete genetic history, is located in our nuclear genome, where the vast majority of the genome exists. And the chance to begin to study that came in the early part of this millennium with new sequencing technologies that allow you to sequence many fragments of DNA efficiently and rather inexpensively. So we've worked a lot on the methods to extract DNA, short pieces of DNA from bones, how you manipulate such fragments to get them into a form that you can feed into the sequencing machines. The machines got much better. We also worked a lot on finding well-preserved Neanderthal bones that have a lot of DNA in them. We focused on a site in Croatia, India Cave, and 3 bones at the time from that site. and then sequenced over 1 billion DNA fragments from those 3 bones and matched them to the human genome that had by that time then been sequenced. To sort of match them, taking into account these differences that are induced by chemical modifications that we had sort of found were common in ancient DNA. So by 2010 we had seen a little bit over half the Neanderthal genome, at least once, so we could begin to ask questions. And one thing we were particularly interested in was this question: what happened when modern humans met Neanderthals? Did they mix or not? And of course, if one would have mixed, we would have expect Europeans today to share more genetic variants with Neanderthals than people in Africa where there had never been any Neanderthals. So we addressed that question in a very simple way. We worked together with a number of population geneticists at the time. We had the Neanderthal genome from Southern Europe there, and we went out and sequenced 5 present-day people with exactly the same technology to have the same error patterns and so on, and did a very simple test. We just start here to illustrate this by comparing 2 people in Africa. We can compare these 2 genomes and find all positions where they differ from each other. We then take the Neanderthal genome and say, how often does the Neanderthal match this African or that African? And it should be 50-50, right? Because there's no reason to think that one African is closer to Neanderthal than another African, because there have never been Neanderthals in Africa. And indeed, that is the case. Statistically speaking, there is no difference in the matching. We then went on to compare a European individual. At the time, we sequenced a French individual because we thought that's an archetypical European person, comparing it to an African individual. And to my great surprise at the time, we actually found significantly more matching to the European individual than the African individual. Even more surprising was that when they took a person from China and compared to Africa, we found more matching. Although most people would agree there have never been Neanderthals in China. Papua New Guinea, even more extreme, no one would claim there have been Neanderthals there, you find more matching. So wherever you look outside sub-Saharan Africa, you find this additional matching. So the idea that came from that was that when modern humans left Africa, those of them that did so, met Neanderthals presumably early on. There were Neanderthals in the Middle East, for example. And if these early modern humans that then became the ancestors to everyone outside Africa mixed with Neanderthals, they could sort of carry with them this Neanderthal component out to the entire rest of the world, also where there had never been Neanderthals. And the result then would be that 1 to 2% of your DNA, if your genetic roots are outside Africa, will come from Neanderthals. What has happened since is that we have developed even better techniques. We now don't have just half of the genome. We have actually all parts of the genome to which we can map these short fragments, and we have studied that 30, 40 times over from a number of individuals who have a few Neanderthal genomes of a quality just as good as any genome you would determine from a person today. And they are in the public domain, and they vary in age about like this. And as we already heard in the introduction, we can then compare these genomes with genomes of present-day people, and you can then more in detail see this contribution from Neanderthals. So for example, on chromosome 9 here, a part of chromosome 9, different individuals, indicating in red fragments that are identical or very similar to the Neanderthal genome. And you will see that different people carry different parts of the Neanderthal genome, but it sums up to 1 or 2% per individual. But you can also jump from person to person and see how much of the Neanderthal genome can you puzzle together in present-day people. And that's at least half of the Neanderthal genome, probably a bit more, is still running around on 2 legs today, so to say, in a fragmented sort of way. And as we heard in the introduction, at this site in southern Siberia, Denisova cave, the archaeologists found this tiny little fragment of the last phalanx of a pinky of a child, from which we could also determine a high-quality genome. And we were very surprised then to find that it was not a Neanderthal, not a modern human, but something else that go quite far back here to a common ancestor shared with Neanderthals about 400,000 years ago or so. So we realized we found this sort of other group of now extinct humans based on simply a genome sequence, not any bones or stone tools and so on. So we called them Denisovans after the site where they were found, and we could ask, had they also contributed to present-day people? And indeed they have. We found no contribution in Europe, but wherever we look in Asia, we find a small contribution, less than 1%, maybe 0.2%, 0.3% or so. And quite interestingly, in Oceania, so out in Papua New Guinea or in Australia, about 5 or 6% of the genome comes from these Denisovans. And not only that, there seem to have been different Denisovan populations that contributed to present-day people. So this is an analysis from another group at Princeton, where they compared genomes of people from Papua New Guinea today, and what's depicted here is sort of similarity to the Neanderthal genome. Identity would be at 1.0 here, so you find fragments on the genome of these Papuans that are quite similar to the Neanderthal genome. In addition, you have similarity to the Denisovan genome here, and you find this contribution from Denisovans that is rather distant from our genome. So it's sort of much more distant from the Denisovan genome than the fragments from the Neanderthal genome. If you now go to Japan and do the same analysis, you find this contribution that you also see in Papuans from Neanderthals. You find a Denisovan contribution here, similar to the one in Papua, but you find in addition a contribution from Denisovans that are very similar to the Denisovan genome here. So there are two contributions from Denisovans here that seem to come from distinct Denisovan populations that were different from each other. So there are at least two Denisovan populations that contributed to people in East Asia, and there might actually have been even more. There are some indications of that. I want to mention sort of one development in the field, and that sort of and illustrate that with this fact that we have so far bone or teeth from Denisovans only from the Denisova cave in southern Siberia. But they must have been more widespread in the past. And we're beginning to see that. There is a site in Tibet, quite high up on the plateau, where a mandible, part of a mandible has been found that is quite old. There might be Denisovan, but one has been unable to retrieve DNA from it. But together with Chinese archaeologists, we have then studied sediments from that cave site. And it turns out you can nowadays extract DNA from sediment samples at sites where people have lived, retrieve DNA from there. Most of the DNA comes from animals that have been in the cave, but sometimes you also found human DNA. So you can then go through the stratigraphy, extract the DNA, and in this case find DNA in 4 of the layers there. And when we sequence that, it's clearly Denisovan DNA related to some of the genomes we have from the Denisova cave and less related to others of them. So I think this is something that will come in the future, that one will routinely retrieve DNA from sediments in archaeological excavations to find out what populations have lived there at what times. So just to summarize the first part then, what we think we know about the origin of Neanderthals and Denisovans from the genomes is that they have some common origin, probably in Africa, and the common ancestor of them come out of Africa and evolve in the west to what we call Neanderthals, and in the east to what we call Denisovans. The border between these groups have lived and probably shifted over time, and we don't quite know about that. But we know that in southern Siberia, for example, the Denisova cave, there sometimes been Neanderthals, sometimes Denisovans. Then modern humans appear in Africa, starting maybe 200,000–300,000 years ago, and they start leaving Africa seriously in large numbers, less than 100,000 years ago, 60–70,000 years ago, mix early on with Neanderthals, continue to spread, mix several times with Neanderthals, and in the east also several times with Denisovans, continue out to the rest of the world where other forms of humans have never been. These other forms of humans disappear, but live on then in this sort of contribution to present-day people of 1 or 2% of Neanderthal DNA outside Africa and 6–7% Denisovan DNA, for example, in Papua New Guinea. You may then ask, are Africans different in not having any contribution from these other forms of humans? We don't know that really, because we don't have ancient genomes of that age yet from other forms in Africa. But since modern humans spread, of course, across Africa too, and replaced these other forms and mixed outside Africa with other forms, it probably happened also in Africa. So I think one will find in the future that we have this contribution also in Africa, but from other groups than Neanderthals and Denisovans. An interesting thing is that we begin now to find very direct evidence of this mixing. The first such case came from Romania, a cave site where cavers found this mandible. It's 40,000 years old, and it looks like a modern human. So it's a very early modern human from Europe. So we were quite interested in if this individual or its ancestors had already mixed with Neanderthals. And indeed, it have. We look on the different chromosomes here and indicate in blue parts that are identical or very similar to the Neanderthal. You find huge segments here sometimes. Sometimes half chromosome arms come from Neanderthals, indicating, of course, that this individual had a close family in its family tree, a Neanderthal ancestor. So you can show that either 6, 5, or 4 generations back, this individual 40,000 years ago in Romania had a Neanderthal ancestor. We thought that this was a unique, that we happened to stumble over this individual. But in the meantime, there is, for example, this site in Bulgaria, where one finds technology typical of very early modern humans. And deep down, there are remains of 3 humans that are around 45,000 years old, among the earliest humans, modern humans in Europe. 10,000 years later, there's also modern humans there. And if we now look at the genome of these 3 individuals at 45,000 years ago, it turns out that they all have sort of close family relatives that are Neanderthals, a few generations back. If we look 10,000 years later in the same cave, there is no special relationship to Neanderthals anymore than any one of us have today. So I think the picture that is emerging is that when we look at the very first modern humans that come to Europe 45,000 years ago, they seem to have mixed really extensively with Neanderthals. And if they were then more numerous than Neanderthals, you could imagine that a big part of why Neanderthals and Denisovans disappeared may simply have been that they were absorbed into larger modern human populations that came to the area. So much of the reason why these groups disappeared could simply have been mixing and assimilation into larger groups. Now, that's probably too simplistic, but sort of may well have been different in different times, different regions, but part of the explanation can be that at least. So for the last part here, I then want to focus on the fact that we now have then good genomes from some Neanderthals and 1 Denisovan, So we can begin to compare it to tens and hundreds of thousands of genomes that we have of present-day people, and of course to chimpanzees and apes as outgroups. So we can then find things where humans and Neanderthals share a genetic change, and that is then goes back to mutations that have happened on the common lineage back here. We can find things that are unique to Neanderthals, that obviously happened since we separated from Neanderthals 500,000 years ago, and we can find things unique to present-day humans that happened on the human lineage. And I want to give you some examples, first with these Neanderthal-specific things and what physiological consequences they can have. The first such example is a gene that encodes a protein that sits in the cell membrane. And this is work by Hugo Seberg, who works in Stockholm, and with us in Leipzig. And this protein is an ion channel. So it's a protein that lets ions come through the membrane when it is stimulated. And in Neanderthals, he found that it carries 3 differences in all Neanderthals from present-day humans. It looks quite different. That's quite unique to have 3 differences in a protein in Neanderthals from modern humans. And this is an interesting ion channel because it sits in the peripheral nerve endings and initiates a sense of pain when you injure yourself. So we then synthesized the Neanderthal version of this channel and the modern human version and tested them in human cells. And for a certain stimulation here then, you find that the Neanderthal version in around red lets more ions through the membrane than a modern human version. So it seems like it would be more sensitive, it would respond stronger to a certain stimulation. You could also show that that's due to that it opens and doesn't get inactivated so rapidly. So it remains open longer for a certain stimulation. Now we thought this was unique to Neanderthals, but an interesting development in the field is also that you begin to have large biobanks, and in Europe it's the UK biobank that is the biggest one, where at the time one had 360,000 individuals for which you have genomic information, medical information, and questionnaires they have answered. So when we looked there, we then found that this was not totally unique to Neanderthals. Some present-day people, 0.4% of people in the UK carry this version of the ion channel inherited from Neanderthals. So we can then use those modern humans as a like for sort of test animals to test Neanderthal versions and study them. So one could then ask on questionnaires that people had answered about pain, all kinds of pain, back pain, stomach aches, headaches, what have you, and just summarize how much pain they would sort of report in their lives. If you just look at what this experienced pain correlates mostly with in your life, it's rather sad. It simply correlates with increasing age. The older you are, the more pain you have in your life. And it's sort of trivial because you have more medical problems. But more interestingly then, carriers of this variant do report more pain in their lives than others. And to sort of give a quantitative feeling for that, if we relate it to this increase with age that we perhaps experience, all of us, it is sort of as if you were 8 or 9 years older in terms of how much pain you report in your life if you're a carrier of this. Now, this doesn't mean that we can say that Neanderthals experienced more pain necessarily. You all know that a certain stimulation may be painful in certain circumstances in a life and even pleasurable in other circumstances. So the sense of pain is sort of modulated very much by your brain and in your spinal cord. Yet it is interesting that people today that then carry on one of their chromosomes this Neanderthal variant, because this is so rare. So we have not found anyone that got this both from the mom and from the father, right? So people who have one copy of this do report more pain in their lives. And Neanderthals, of course, had this on both chromosomes. So So maybe we sort of have to reconsider our view of Neanderthals as these sorts of insensitive brutes. Maybe they were actually too sensitive and everything hurt when you met modern humans, and so they became extinct. The other example I want to mention to you then involves a hormone, progesterone. You're probably familiar with it. It's sort of a hormone with many functions in the body, but the primary function is to be produced in the ovaries after ovulation to prepare the uterus for a possible pregnancy. And we are now interested in the receptor for this progesterone hormone. So to which the protein to which a hormone binds in its target organ. And that progesterone receptor has a variant that was already known and existed, as you can see here, outside Africa. So it's not present in Africa or hardly at all. It is present outside Africa. And whenever you see a genetic variant with this distribution, it almost always comes from Neanderthals, actually. And it's turned out that this Neanderthal version is associated with an increased risk of premature babies, of having preterm births. So it had been speculated that that might be a sort of risk to Neanderthals because of having premature babies is, of course, a risk to the baby, particularly when you don't have any medical care. But an interesting development in the field is also that we're beginning to have tens of thousands of genomes of modern humans of different ages. Sort of over the past 15,000 years, we have now something like 20,000 genomes. So we can study the frequency of a variant like that over time and see if it has increased in frequency or gone down in frequency. So we can do this, for example, for this progesterone receptor variant now. So if we start here 15,000 years ago and go forward in time towards our time, and black are now carriers of this Neanderthal variant, and gray are non-carriers. The red ones are simply the Neanderthal and Denisovan genomes. So we now move forward in time here, and you will see around 7,000 years ago that the frequency of this almost explodes, which seems to make no sense whatsoever, right? If you have sort of increased risk of your baby actually dying, why would this increase in frequency? So we went back to the UK Biobank and looked what these variants of the progesterone receptor are associated with in present-day people. This is unfortunately a little backwards. Now we look at what the modern version of this allele, so the non-Neanderthal variant, is associated with in the UK Biobank. And we find that it's associated with bleeding early in pregnancy. It's associated with increased risk of miscarriages, and actually negatively associated with the numbers of sisters and also brothers that you have. So it actually seems that the Neanderthal version sort of protects against miscarriages. So the story seems to be that, yes, this Neanderthal version is associated with an increased risk of premature births, but it's also protective against miscarriages earlier in pregnancy and actually results in more live births. So it seems to be an evolutionary trade-off, so to say, that this variant saves pregnancies that would otherwise result in miscarriages, and the price you pay is an increased risk that these babies are premature. And we begin to understand why that is also, because if we look at how much of the progesterone receptor you make if you carry the Neanderthal version, you actually make more of the receptor. You have more of the receptor in your target tissue. So you could imagine that you have more of a progesterone effect then when you have more receptor. And indeed, in the last few years, there have been two big studies where one has given progesterone to women who has experienced several miscarriages and have substantial increase of live births when you give the hormone, compatible with the idea that more progesterone effect sort of prevents miscarriages. And this is sort of a pattern that many medically important variants that are already known turn out to come from Neanderthals. Just a little example are two enzymes expressed in your liver that clearly come from Neanderthals, and they are involved in digesting many drugs we take. So for example, if you take ibuprofen against pain and are a carrier of the Neanderthal version, you have a much better effect because the half-life is much slower. You have much more of this in your blood a couple of hours later than if you have the modern variant. But more seriously, if you take warfarin against blood clots, if you have an artificial heart valve or other blood clot problems and carry the Neanderthal version, you need to take a lower dose because you have more of it in your blood and a bigger risk for complications of bleeding. Another example of sort of influence from Neanderthal versions came with this pandemic that we are hope we are come through. As you know, if you're infected with the SARS-CoV-2 virus, most people have very mild symptoms or no symptoms at all, but some people become severely ill and some even die. And we know many of the risk factors, old age, for example, male sex, diabetes, what have you. But all these risk factors are not enough to explain why certain people become so very ill and die and others have hardly any symptoms. So there must be factors in the host too, in the people who get infected. So we were peripherally involved in a big international consortium back in 2020 to study genetic risk factors for becoming severely ill. And an early result in the first summer of that consortium was that there was one major risk factor on chromosome 3 here that had a big influence on how sick you get if you're infected. And when we then looked at those risk variants there in a tree like that, we almost fell off our chair when we looked at it, because it turned out that these risk variants are very close, particularly to one Neanderthal genome we have there. Whereas all the protective variants are not at all related to Neanderthals. So this risk factor for becoming severely ill had clearly come over from Neanderthals and exists in some people today then. And it's quite a big risk factor, so if you were admitted in Scandinavia with severe COVID to the hospital early in the pandemic, and were not a carrier of this variant, you had a 7% risk of dying. But if you were a carrier of this Neanderthal version, you had almost a double the risk of dying. So there's of course a big interest in what is in this region on chromosome 3. We and others have worked hard on it, and unfortunately it's a very complicated region with many genes. It would be great interest to understand why this confers risk, because we could then perhaps treat severe COVID better. But at the moment there are sort of as many theories as genes almost in that region. But it's also interesting to look at the distribution across the world of this variant. It has this typical Neanderthal thing. We're not seeing it in Africa and seeing it in other parts of the world. So from the frequency, we can estimate approximately how many extra deaths have we had in the pandemic due to this Neanderthal contribution. And it's clearly over 1 million extra deaths, probably more, approaching 2 million actually. But you can also see that the distribution outside Africa looks very interesting. So it's sort of absent in China and Japan, in East Asia. It is quite frequent in India and Pakistan, Sri Lanka. About 50% of people are carriers there. This clearly indicates that it has been selected against in East Asia. We don't know why. Perhaps coronavirus epidemics in the past have selected against this. But it has clearly been positively selected in South India. It has been good to carry this variant there. So we don't know what these factors were, but we're beginning to learn that this region has influences other than influencing your risk in a pandemic. So Hugo again noticed that if you look at this region of chromosome 3 and go 1 million base pairs away, there is located a gene, a well-known gene that some of you may have heard about, CCR5, which is a co-receptor for HIV. So the virus that gives you AIDS uses this protein to get into the cells. We know that people who have a deletion of this on both their chromosomes, so CCR5, are actually resistant to HIV infection and cannot be infected. And when we now look at carriers of this risk variant for severe COVID, we find that they express less of CCR5 in their tissues. And indeed, if you look in cohort studies at the risk of becoming HIV positive if you're exposed to the virus, it's reduced by about 25% if you're a carrier of this. So the situation is that this Neanderthal segment on chromosome 3, it increases your risk for severe COVID if you're infected with SARS-CoV-2, but it decreases your risk of becoming infected with HIV if you're exposed to that. So this illustrates that I think that almost every genetic variant we study have actually different effects depending on the environment, depending on what virus you're exposed to. So it's a double-edged sword, and clearly, you know, what have changed the frequencies between East Asia and South Asia is not HIV or SARS-CoV-2, which are just there for a decade or a few years, but other things. It's actually a multi-edged sword. Before we leave Neanderthals and the pandemic, I should just say that when one now does bigger studies, you find other parts of the genome that influence the risk of having severe COVID. On chromosome 12, for example, there is one Neanderthal variant, and in this case it's actually protective. In this case it's actually good to carry it. In that case, we also know what it does. It degrades double-stranded RNA, the sort of this virus. But unfortunately, the effect size is much smaller. So the risk on chromosome 3 about doubles or triples your risk of dying. The protective variant on chromosome 12 decreases it by about 20-25%. Towards the end, then, I want to mention the other area that we're particularly interested in our group now, are those changes that are then unique to modern humans. And we are so interested in them because we think that there might hide sort of some of the functional backgrounds of being a modern human from a biological perspective. And modern humans are, in my opinion, special. There are certain things that come with modern human when we compare ourselves to Neanderthals, Denisovans, other forms of humans that were there before. With time, at least, the technology starts changing rapidly with modern humans. We see that, for example, in that it becomes regionalized. An expert can immediately say if early modern human technology comes from Central Asia or from Western Europe, whereas Neanderthal technology was pretty homogeneous over their entire distribution, shifted a bit with what type of stones were available. Figurative art that really depicts something, comes only with modern humans. There are things that are probably Neanderthal art, but when I get a little bit sort of facetious, I would say that Neanderthal art looks like very modern art to me, because I can't see what it depicts. Whereas art that really depicts something like this comes with modern humans. And of course, modern humans become very numerous, spread to all parts of the world, across open water and so on. So we can sort of list all these things now that happen on the line to modern humans and exist in almost everyone or everyone today. And that list, that catalogue of things, is not that big. It's something like 30,000 changes in our genome. So as I said, two genomes between two people in this room differ by about 3 million differences. But if we make the requirement that everyone today, or almost everyone, should carry something, but Neanderthals not have it, then we're just at 30,000 changes. And if we focus, for example, on changes that change an amino acid in a protein, it's just around 100 such changes. And we have a bias, of course, that we think that modern humans, that's something with how the brain functions that make us special. Neanderthals had just as big brains as we had, but the idea is that somehow cognitively there is something special with modern humans. We have this bias. So we've been I just want to give you a flavor for what we work on over the last few years and where we are now. We sort of can look, for example, on the catalog of all these genes that have amino acid changes they encode that are of this category. And something that struck us there was that 3 of these genes encode proteins that have to do with how the chromosomes are pulled apart when a cell divides. It seemed very weird to me that something so basic in the cell might have changed recently in human history. So they're involved in sort of how the chromosomes are pulled apart when 2 daughter cells are formed. But it reminded me then of work we had done earlier, where we had used brain organoids. You can take stem cells of humans or of chimpanzees, and in the tissue culture make little balls in tissue culture that sort of mimic the early steps of brain development. And you form this epithelium where cells are formed, and this is worked together with Wieland Huttner's group. And this is then comparing humans and chimpanzees, how these stem cells that form neurons are formed, and what is stained here are the chromosomes with the DNA. And if I play this little movie, you will see here how the cells, how the chromosomes line up to the right there, here, and are pulled apart there, and then pulled apart there. And you will see that this happened a little earlier in the chimpanzee cells than in the human cells. And indeed, that's something that we had already found, that sort of the time when the chromosomes line up and are checked against each other before they are pulled apart is shorter in chimpanzees and in humans. So that made us then think maybe this has to do with these changes where we differ from the Neanderthal. We sort of put these changes in these 3 genes into the mouse, actually, so the human form, comparing it to the Neanderthal-like mouse form, did these experiments. And indeed, we could see that with the changes in the 3 genes, we had a longer such phase where the chromosomes lined up. We just changed it in 2 of the genes, we also had it. Singly, we did not see it clearly. So we focused on these 2 genes, put in the Neanderthal-like changes in these brain organoids again, and indeed, we see then a shortening of this phase here. So this difference then between humans and chimps that observed have happened since we separated from the Neanderthals. And it seems to perhaps have some effects, because if we look on the number of chromosomes that sort of missegregate and are not divided between the cells properly, it increases when we have ancestralized the cells, made them Neanderthal-like. So there seems then to perhaps be more accurate chromosome segregation in modern human brain development than Neanderthal brain development. Now, the big question is, does that have any consequences for the adult individual? Because we're looking here at things that happen early in development, say maybe in the first 2-3 months of development, that may well be compensated for later. I just very briefly mentioned one more such change in an enzyme in energy metabolism that we similarly put into Neanderthalized these organoids. And when we then look at these stem cells here, we find less stem cells in the Neanderthal-like organoids. That actually results in less neurons being done. So the big question at the moment, one of the big questions for us is, does neuron formation in modern humans differ from Neanderthals? We don't know, really, but some things to consider is that, as I said, this happens early in development and might be compensated for later. So it may not have any consequences in the adult. Another consideration is that most features in a human are influenced by many different genes. We already saw a hint of that. We needed to change 2 genes to see the effect there on how chromosomes segregate. So it may be that there are other changes we haven't studied yet that sort of compensate for these things we find here. And the third thing we should consider also is that for almost all of these, not all of them, but many of them, we do find present-day people who carry the ancestral version as a result of gene flow from Neanderthals and Denisovans. So I think if we want to have some kind of a definition from a biological genetic perspective of a modern human relative to Neanderthal, it must clearly be a sort of combinatorial thing, where we would say that you're a modern human if you carry many or most of these changes in that catalog, but any single one of them is not at all necessary for sort of functioning well as a modern human. But that's sort of where we are with our thinking with these things. So I hope I have then convinced you that having the genome of our closest evolutionary relative is interesting, because we can look for these things that are unique to modern humans, can look for things that are unique to Neanderthals, we can sort of study the contribution of Neanderthals to present-day people. And we do that by using these big biobanks that are becoming more and more important, but also by doing experiments using stem cells and brain organoids, for example, and sometimes even humanizing and Neanderthalizing experimental animals such as mice. And I should say that this work then goes on in Leipzig in Germany in the group there, and to some extent also in Okinawa in Japan in a small little group there. And with that I then thank you for your attention.
Merci infiniment, cher Svante, pour cette passionnante conférence avec beaucoup de perspectives très nombreuses sur la médecine, les liens entre humains et non humains, les épidémies, l'archéologie, l'usage de l'archéologie, de l'histoire. Je pense qu'il y a beaucoup de choses à dire. Et dans cet esprit, j'ai décidé avec mon équipe de poursuivre la discussion en demandant à des chercheuses et des chercheurs en sciences sociales de l'EHESS de te répondre, to answer you, dans les prochains mois, pour voir la manière dont des chercheuses en sciences sociales, dont vous avez les noms, je crois, dans le programme, et vous avez la suite de le cycle de conférences Marc Bloch, qui vont répondre sur les points que tu as évoqués, la question médicale, les liens entre humains et non humains, le rôle de l'archéologie, j'espère oublier personne, les chimpanzés aussi, et la manière dont l'histoire a considéré à l'époque des Lumières les chimpanzés. Donc je vous invite toutes et tous à venir à ce cycle de conférences. On vous enverra bientôt le programme définitif, les lieux et les moments. Ça débutera en janvier. Et l'idée est vraiment d'ouvrir ce débat interdisciplinaire. On peut être d'accord, on peut ne pas être d'accord. Mais je crois qu'il est essentiel qu'une institution de sciences sociales ouvre le débat et le fasse vivre, puisqu'on voit très bien que les questions qui ont été posées par Svante sont des questions absolument essentielles pour l'avenir de l'humanité et de la science. Merci à nouveau Svante d'avoir été parmi nous. Thank you so much for being with us. It was a real pleasure to have you here. Thank you.